Welcome back to this afternoon invited talk section. We have 3 invited talks. Our first speaker is Roarke Horstmeyer from Duke University. He's an assistant professor in biomedical engineering and electrical and computer engineering. And he's also a scientific advisor for Ramona Optics, and very well known for his work in tychography, microscopy, and cameras. So, thank you for coming.
Okay, great. Thank you. All right, nice. Okay, so yeah, this talk is about some of my lab's work and also work at Ramona with making arrays of cameras. So it's a pretty simple talk, so it's hopefully enjoyable. I have some cool videos. But the problem we're trying to address when we are creating these arrays just surrounds the historical development of microscopes and how they really haven't changed very much. So here are 2 microscopes from Zeiss. You can look at other companies that make microscopes, but over the last couple hundred years, they're pretty much the same technology. They have an objective lens, something to look through, to see through the objective lens. And then what's new on the microscopes today is what that black thing on top, it's a digital image sensor, right? So those are typically slapped on via a beam splitter, and you get also digital images, right? And so the issue that the microscope objective presents is it kind of limits the field of view you can study when you're looking at high resolution. And so that makes looking at large samples a really slow process. You have to scan around step by step. And it makes looking at live samples challenging. And so really what we're trying to address in a lot of my lab's work is looking at much larger areas synchronously. So you can study at high resolution over large areas new phenomena. So why is that important? And so there's a few different reasons why that's important. There's the desire to see freely moving model organisms, high throughput experiments, something called cytopathology, which I'll explain quickly later, and then this area of microsurgery. So I'll show examples of each of these. These are areas where we've learned it is really important to see over a large area at high resolution. So how do we do this? Like I said, we're using arrays. So our first step is to shrink the performance of a microscope down into a small package. That's not surprisingly doable, given we have the performance of almost DSLR type imaging in our pockets, right? The same thing can be done with microscopy. Then we put these microscopes in arrays. And so in a standard configuration, the configurations we started out with, we made the field of view of each of our little microscopes just abut or slightly overlap its neighbors. So that camera 1 would just see the top left corner of the scene, camera 2, the next adjacent area, et cetera. And then we would stitch all those images together to create large area, high resolution composites. So this idea of arrays or multi-aperture imaging is certainly not new. This is by far not an exhaustive list of technologies. but I think many of you in this room are aware of a lot of these. So we have the famous Stanford Light Field, camera array, miniaturized versions. There was a company originally named Pelican Imaging, and it kind of changed forms a few times. Maybe some in this room have experience with that startup that were creating small arrays for smartphones. Arrays are used in astronomy, and they're in our pockets. And then there's these multi-scale lens designs where you can have primary optics in front of arrays. And I'll show a couple examples of that. Where we're focused is sort of in this what we call mesoscopic regime. It's not quite where the really high resolution microscopes are. There's excellent technology there, and it's not in the macro, large area imaging regime. It's in that middle space. And there's really limited actual 3D technology. So capturing depth at tens of micrometers resolution is challenging. And so a lot of the work I'll show here is doing large area, high resolution 3D imaging. And we're moving off that kind of traditional trade-off between field of view and resolution and trying to give more pixels. Okay, so our first array looked something like this. This was our Gen 1 design. It had 54 synchronized image sensors, each one at 13 megapixels. We could run it at up to 240 frames a second. At full resolution, it was 700 megapixels per frame. And so it's a lot of data, and so we've also naturally been working hard and getting good at handling all of that data streaming in at 5 to 10 gigabytes per second. Okay, so cool video number one. Here's an example of capturing at, I think around 100 frames a second carpenter ants as they're freely moving around. We slightly overlap the cameras so we can use stereoscopic or rather photogrammetry methods to get 3D height as well. And so here's this, very coarse, rough 3D height, I'd say. We're still working on refining it. Measurement of those movements of those ants so we can watch their limbs moving up and down and antennas moving up and down. A really large application of this is in high throughput imaging, and that's a fancy way of saying in biology where biologists want to image lots of things and then use those data sets to derive insights. The vehicle to do high throughput imaging are these plates in the back. They're called multi-well plates. They're 8 by 12 centimeter dishes that contain any number of wells between 24 up to 1536, small little places to put your sample. And so a lot of our first research efforts were focused on looking at zebrafish. So these are studied at high throughput. They're a model organism. They're tiny, several millimeter sized fish. They're transparent when they're young. And they're basically the simplest vertebrate you can work with. And so a lot of early drug discovery toxicology studies center around the use of these zebrafish to gain insights. So our array can record video of 24, 96, et cetera, of these zebrafish during interesting behaviors like hunting. So you can see tiny little black dots moving around. That's the food that they go and capture immediately after they exit their embryonic stage. And naturally, it's a lot of data, so it's a great task to apply data analysis techniques to. In our work, we've been developing these statistical measures of what are called ethograms. These are behavioral categories. And then we can just plot these behavioral categories for each well, for example, in that 96 well plate. And yeah, a lot of our collaborators are really enjoying this and gaining new insights into behaviors that are associated with seizures or immunotoxicology effects, things like that, that otherwise weren't possible without this imaging system. And so it's not so much just the imaging system, right? If you just provide that imaging system, you'll flood these poor biologists with data. You have to also solve the coupled problem of what do you do with all that data. And so this is just one of those examples. Okay, so another configuration we can image in is zooming in more so the fields of view don't overlap, and then you just have a bunch of microscopes imaging in parallel effectively. So our Gen 2 design was more compact. We squeezed more image sensors into a smaller area, so roughly the size of a credit card. We have 48 microscopes, and we can put 2 of those together to make a 96 array. And then these are kind of modular systems. We had one upstairs demoing yesterday, if some of you saw it. And so we have a bunch of these lying around. I'm happy to share. If you're interested in grabbing one of these modular arrays, just let me know. I'm happy to chat about that. And so we're putting these into high-throughput drug discovery applications, like I mentioned, with the multi-well plate. Big area right now that are using or studying these specimens at high throughput are with organoids. These are small organs that rather small multicellular clusters that recapitulate your organ functions. So you can take a single cell from patients and derive brain organoids. So that's neurons firing. They're fluorescently labeled. There's a cardiac organoid up in the upper right. You can see it's beating very slightly. And then there's tumor organoids, naturally, for cancer, cancer research and cancer treatment. We're also applying our microscope to look at slides, not just these well plates. So a large category of the workload of a pathologist is to look at cytology slides. These aren't thin tissue sections. It's kind of a material that's taken from a needle that's extracted from sites of interest to look for small, warning signs. It's kind of the first modality pass when folks are examined for cancer. That's traditionally hard to image, and because the specimens are three-dimensional. We're also doing some quantitative phase imaging with our imaging system. So the way we're doing that is with illumination that allows us to capture different, images in different illumination conditions, I guess is the best way to say it, from our parallel microscope. And then we have to take multiple images, and we combine those multiple images per field of view into a quantitative phase reconstruction. And these are some of the patterns we're showing, and this is what some of our phase results look like. And so we're doing this in live cell imaging contexts, right? Because we have all these microscopes just sitting there, we can capture an image every 30 seconds to watch these cells as they migrate and interact. And so a key target of this is to look at T-cell tumor cell interactions. T cells are moving rather quickly. They're notoriously hard to image at high throughput, but here we have 48 or 96 microscopes sitting there. We can just stream all that data and gain new insights from these interesting cellular dynamics. Okay, so I'll end with what else can we do with these systems? The first thing that we had laying around was this giant lens, and so we put it in front of our array to make what's called a Fourier light field. This is essentially, it's easiest just to understand what a Fourier light field is by looking at the data. So here's a picture of a small dinosaur figurine captured with this configuration. You just see it from different angles, right? Just like a normal light field. Each of our cameras sees the same thing from a different perspective. And so we have 48 13 megapixel images, all from different angles, and naturally we can put those together into 3D composites, right? And so the application of this is, I think, a great application of this is in surgery. So we're working with surgeons to use our 3D imaging technology to help them with their tasks that require a microscope. And so the catch term for that is microsurgery. And so I'm not going to show gross surgical videos, but you can kind of judge from these videos of just skin and a dried rat skull that it's suitable technology. And just resolution-wise, we get pretty high resolution. So we can zoom in and see the ridges of your fingerprint, for example. So it's in the tens of micrometers height resolution regime, which is suitable for a lot of applications in microsurgery. All right, so naturally when you're thinking about 3D imaging and cameras, you'd want to have this configuration, right? You'd ideally curve your imaging systems around the object of interest. We thought about and tried for a while making a curved array of synchronized cameras, and getting that to be over a dozen cameras or so is tough. And so we ended up not doing it, but instead, Kevin, who's now starting a lab at Michigan, had a nice idea of just putting a parabolic mirror beneath the array and focusing the lenses at infinity, and the mirror acts as the second focusing element, and you essentially get like a 4F type configuration. But you can get very steep angles looking at your sample of interest. And so an example snapshot from that setup is shown here of a zebrafish that's fluorescently labeled. And then we can kind of put all that data together into a volumetric video of that, whatever's in the focus of that parabola doing its thing, but at high frame rates. So we can go up to, I think, 240 frames a second with this 3D volumetric imaging technology, which is unique. And so we're beginning to image some fast biological phenomena in this new mirror bowl setup, I call it. Okay, that's it. I'd like to thank you all for your attention.
We have time for questions.
So, immediate question, after having lots of cameras in one system, how do you get data off them without microwaving your computer?
Yeah, great question. And so I don't know why this keeps going. So yeah, we all of the cameras are on a single PCB, and then they route data to a single FPGA. And so that is critical for heat management. The heat is taken away. The FPGA, then we can throttle the data going to the computer. And so we can plug it into a laptop that'll throttle the data appropriately. We can plug it into a big desktop and go up to 10 gigabytes a second. And so basically, like, that's a lot of the hard work. And so it's optimized to work on Linux, but it can also work, on other operating systems. But we had to get into that firmware to make that happen. Yeah.
Any other questions? I have one. How can you improve the resolution? You said you were trying to be in the middle. Can you use this large array but still get better resolution?
Yeah, great question. So that those 3 slides I showed, the cytology application, that's at like a little less than 1 micron resolution. We've made little lenses that go down to 400 nanometer resolution, so like a 0.5 NA. You can go further, but eventually, like, you'll need oil, and then you have an array of all these microscopes with oil. I don't want to deal with that personally. But yeah, and then you can do some structured illumination or other kind of resolution enhancements techniques, like Fourier tachography. Those are all on the table. Sure. Yeah.
Any other question? I have another one.
Okay.
With the last configuration, can you do tomography?
Yeah, so we're doing tomography.
Okay.
Yeah, so that's a full, this is a full volumetric reconstruction that Kevin created. It's on the order of, 800 by 800 by 800 voxels.
But is that diffraction tomography? Or?
Oh, no, this is fluorescence. Yeah, so we could do diffraction tomography. Yes, we have not done that.
Okay, listen the speaker again. Our next speaker is Matthew O'Toole. He's an associate professor at Carnegie Mellon in the Department of Computer Science and Robotics. And he has a PhD from, give me a second. He was a PhD from here, actually, the University of Toronto, and a postdoc from Stanford University. And he has also an NSF Career Award and an impressive number of Best Paper and Best Demo Awards at CVPR, SIGGRAPH, and ICCP. So thank you for the talk.
Great, thank you. You mind, Raphael, if I borrow the mic for a bit? Yeah. Thank you. All right. I'm going to start things off with a demo. I'm going to be talking about spatially varying autofocus. And so I figured, let's just give me a second here. All right, so what you're seeing here is basically a stream of a camera that I have up on stage of this object. And right now, it's behaving like a normal camera. The focal plane's over here somewhere where the object remains in focus. If I move things in front or behind, things get blurry, right? So let me place the object right here. I'm going to turn our camera into a mode that allows us to create all-in-focus images. So here we go. I have an all-in-focus image. Not very interesting because it was already in focus, so let's just move things back. It's in focus in the center of this image. So I've actually told my camera to control the focus of specific pixels or regions within the scene such that the center of the image is now in focus, and the focus setting for all other pixels is actually back at that original plane. I can form an all-in-focus image by setting another mode where now if I were to place other objects into the scene, like so, it's now trying to get the focus settings that brings these two objects to sharp focus at the same time. Comparing that to a conventional camera, you'll see that things can become a lot blurrier. And I'll be talking about this today. Okay. Hand this back to you. Thank you. All right. Great, there we go. Okay, we can skip that. So we're focused on taking scenes like this, which has a very narrow depth of how to focus the scene into forming an all-in-focus image. And we're going to do so in an interesting way, hopefully you'll find. Here are a few other examples that compare conventional imaging to our all-in-focus images. And one of the things to note here is that these images were formed optically. That is, we're reading out basically the data from the sensor themselves, not doing any post-processing on it. So the result of our all-in-focus images is due to how we focus light onto the sensor, not due to any computational processing that's applied to these images after the fact. So before I get started, of course, I want to acknowledge my two collaborators, Yingxi, who's a student of Ashwin, and myself. She couldn't be here today. She will be hopefully presenting this work at ICCP in Hawaii. So if you're going to be there, I'd recommend that you go and chat with her about the work. And we're going to try to bring an updated version of this demo over there as well. So this is basically a culmination of two works. So we presented some work back in 2023 on something known as a split Lohmann multifocal display. So it's a VR headset, well, a VR display at the very least, that made use of a special lens. And so we're basically using this lens for imaging now. So I'm going to focus also on how we can get this lens to form things like all-in-focus imaging or images, as well as maybe tackle a few other imaging problems along the way. All right. So we'll review basically a little bit of this prior work, which basically starts off with something known as a Lohmann lens. Some of you might know this as an Alvarez lens as well. So it consists of these two optical elements that have a particular profile associated with them. And when you pair them up together, it kind of acts like a lens that has the combined thickness of these two elements. So if I were to sum up the thickness of these two elements, it basically produces something that looks like a piece of glass. So it doesn't really do anything by itself until you start shifting these two lenses relative, or two elements relative to one another. So when I do that, the combined thickness now starts to come up with some other shape. So you have this convex lens now, and we can evaluate what the profile of this lens might look like. And what we'll find is that one of the terms here describes basically the curvature of this lens, where displacements between these two optical elements will create different shapes for this lens, changing the effective power that of the combined elements has on an imaging system. We can also translate them or the other way, producing concave lens, and the amount of translation between these two elements determines the power of the combined elements. So if we want to use it for focusing, what we might want to do is, for example, build a 4F optical system so that you have a sensor on the left-hand side and maybe a target on the right-hand side, or a focal plane on the right-hand side that we're imaging, and then we can drop this Lohmann lens in between. And if we want to change the focus here, we can just slide these two elements relative to one another. Now, the effect of this is that you end up changing the focus globally. We're interested in locally controlling focus. So this is not going to work for us. Instead, what we're going to do is change this optical system somewhat. We're going to create two 4S systems. So here we have two image planes and our sensor on the left-hand side. And then we're going to take these two elements and put them in different parts of this system. So we've more or less recreated what we had before. It's just kind of in a slightly different optical configuration. For to move these two things relative to up and down, in this case, it's going to change focus like before. But this particular system has an advantage in that we can, instead of mechanically moving these cubic phase plates, we can drop in an SLM in between that will instead steer light by a small angle in the very center. And the effect of doing that is that you'll end up changing focus. So we can, instead of moving things around, we can just apply a pattern on the SLM that steers the light a little bit to control the focus settings of this imaging setup. The second advantage is that, well, the light that's reaching this pixel is only passing through a small portion of the SLM. So I can actually do this in parallel for other pixels as well. So in this case, we have different pixels. We're steering the light at the SLM by different amounts to control the focus of each of these pixels independently from one another. Okay. So the SLM patterns I'm not going to go into in too much detail other than to say these basically it's just a phase ramp used to steer the light at this particular plane. And this phase ramp is basically derived from some depth map that determines the focus at a local region within our sensor. So this is what the optical setup looks like. Here on the top, we have our objective lens, which will form an image at some plane. And then we have our first 4S system, mapping that light to a spatial light modulator. It's passing through a cubic faceplate as it does that. Here, we're working with a reflective SLM, so light actually goes back through that same cubic faceplate and makes its way to our sensor, and it informs our image. So in this case, we might have a conventional image. We were to program our SLM with the right depth values, for example, a depth map of the scene. We can then form an all-in-focus image. We can even compare this to an image where we have a small aperture. So we took a separate camera, shrunk the aperture down as small as possible, took an image, and you can see that in certain areas, for example, over here and over here, well, there's a clear difference between the details in those regions because the depth range here is pretty extreme. You have things in the foreground that are very close to the camera, and in the background is maybe 3 meters away. So the second part of this, however, is that we need to figure out what is that depth map used to form those all-in-focus images, right? So that's not given to us. We have to figure that out somehow. And so the idea here was, well, let's just look at what conventional cameras do. So conventional autofocus, well, looks at what happens in a particular region of the sensor, and it can do a number of things there. But basically, it's using that to derive what is the right focus setting for your particular lens. In our case, we want to basically mimic that type of operation, but for lots of different patches across our entire sensor. So there are two ways that cameras typically do this. The conventional way is to use contrast detection autofocus, or CDAF, which will try to search for the focus setting that maximizes contrast. Another way is to use something known as a dual pixel sensor to implement phase detection autofocus, or PDAF, which splits the signal into two, forming basically a stereo pair of images that can be used for focusing. So we'll touch on these very briefly in case you're not familiar with this already. So here's a video from Sony that they had, which you have a subject coming into foreground, and then it's demonstrating how CDAF works. So basically, you're trying to maximize the focus, or contrast rather, at the bottom of the image. And you do that by basically sweeping the focus setting of your lens in order to maximize contrast. And we can do that as well. So we, unlike conventional lenses, because we're just displaying patterns on the SLM, one of the advantages that we have is that we can kind of jump to different focus settings. So it provides a little bit more flexibility in terms of how we implement CDAF algorithms. We do a course-defined search across multiple different focus settings for every patch independently. And in our case, instead of just focusing on one plane, as with a conventional camera, we can bring all points in the scene into focus at the same time. And in the top right here, you have the depth map derived using this technique that we're using in this case. And here we're comparing conventional photos with one captured using CDAF. So another approach is using phase detection autofocus. I'm using a version of that with this demo here. And if we're to look at that video from Sony, once again, with CDAF, you have to kind of sweep that focus and find the right focus setting, and it takes a little bit of time. PDAF tends to be quick. All right. So it tends to jump to the right focus setting right away. The idea here is that you use a special type of sensor, one that offers you, has a dual pixel image formation model associated with it that basically splits the image in two. So you form two images effectively, one with the light passing through the left side of your aperture, your lens aperture, and then one forming an image with a light passing through the right aperture. So the left and right images are shown here on the right-hand side. And when something's in focus, well, they should line up perfectly. When things are defocused, either behind the focal plane or in front of it, you're going to end up having a disparity whose sign and magnitude will determine basically what direction you should be focusing in and how much. So using dual pixel images like the one shown here, which is captured using a Canon R50, I believe, can be processed in order to recover a depth map that can snap the focus to form an all-in-focus image very quickly. So in contrast to a conventional photo, PDAF also produces pretty nice results a little bit more quickly than the CDAF version, but requires a dual pixel sensor in order to do so. So we did some analysis on how the PSNR changes with the number of captures. Phase-based autofocus tends to work best here. If you have enough time, you can run a focal sweep and or capture a focal stack and process things as well. One of the advantages with the phase-based autofocus and contrast-based autofocus, though, is that once you have the right focus setting for the scene, then the next frame that you capture, for example, in a dynamic setting, you already have formed your all-in-focus image. So that's a clear advantage over some of these other techniques, like capturing a focal stack and then processing that. You can also do other things other than all-in-focus imaging. So for example, here we might be interested in getting rid of this grid-like structure right behind Luffy. And we can do so by just focusing on the background instead. So we can provide it a depth map or to control the focus such that, well, you just blur out that grid pattern or a grid structure so that you only have the subject in the foreground and the background in focus. Here we have a image of Paris where it's been tilted, so that's why there's a lot of blur here in the foreground and background, or what looks like the foreground and background. If we're to measure the depth of the scene, we can form an all-in-focus image, and you can also play around with focus settings as well to produce some other interesting artistic effects where you have certain subjects in focus and other things out of focus. So I've already shown you a demo, but assuming I have a little bit of time left, and I didn't see anyone flag me yet. Okay. I got flagged. Basically, here's our conventional image, all-in-focus image. We're removing one thing relative to the other object. But I'll leave it at that. So, of course, there are a number of limitations and challenges that still remain. One of the things I should mention is, well, we have this giant light source up front. It's because this system's not particularly efficient. It's as efficient as 12.5%. In fact, this is even less efficient, maybe 5% throughput. This is due to polarizers and beam splitters in the setup. I think that this is surmountable. There's no hard constraint on we can make things a lot more light efficient. It's just a matter of some optical engineering. There are some other issues. For example, the SLM isn't perfect. There's some amount of unmodulated light, so there's basically a blurry signal that's overlaid if the object that you're imaging is far from the focal plane. We can potentially remove some of that using some computational post-processing. There's challenges around depth edges. The thing is pretty bulky. So some of these things are harder than others. For example, shrinking this down is not so easy. But there's some interesting challenges, and I think that there's still work to be done on this, and so check back with us in a couple of months when we're presenting this at ICCP if you want to see an updated version. So that's it. One thing I'll mention is that you cannot go to our website to get access to the paper. It's not there yet, but hopefully we'll have it up in a couple of weeks. And again, I want to acknowledge student Yingxi and Ashwin for their hard work on this project as well. Thank you.
We have time for questions.
If you have any insights on how to make it less bulky, I guess for both systems, the display and the camera.
Yeah, well, if someone has insights, they can share it with me as well. We've discussed it a lot. The biggest issue is the fact that we have these One way to get around that is to try to fold the optics, and we do that to some extent here. So the illustration that I had was all basically, we might be able to fold things even more. But yeah, it is a definite challenge. There are certain applications where bulkiness doesn't matter, however.
Kiitos.
So, I mean, the split lumen lens is basically a proof of existence that there's an optical system that allows you to do sort of per point focus. Are there others? Other solutions?
Yeah, so one question, so we took basically the split lumen lens using some simple phase ramps on the SLM. One thing that we haven't explored, however, is how to, for example, even with those optical elements that have a particular profile, how to optimize the SLM patterns. We haven't dug into that too much. I think that there's some benefit there. And then there's a question as to how to jointly optimize everything. So there are definitely things that we could potentially do on the optics side. There's definitely things that we can do on the computational side as well to kind of clean up some of the images. As I mentioned, some of the light is unmodulated, but it's potentially possible to kind of identify what that blurry component is and subtract that out from the image. So I would say yes, but I don't have any examples to give to you right now.
Yeah, it's a great talk, Matt.
Thank you.
There's no fundamental reason you couldn't also use dual pixel, right? I mean, the dual pixel disparity is not that high. So I think right now it's just your setup. You don't have a dual pixel back, but you could incorporate dual pixel technology into this, right?
So we do have a dual pixel thing here.
Yeah, okay. That's what was making it so fast.
Yeah, so
Yeah.
it's...
So you've got the calibration to work.
Yeah, it works. One of the things I'll note, however, is that, unless someone can correct me, I'm not aware of any machine vision dual pixel sensors out there. So we used an R50 Canon camera to capture dual pixel
That's probably
images,
right.
but
Probably
it's
not
really
one that
slow.
gives you access to the
And
firmware.
we hacked something up together to produce dual pixel images here.
Cool.
Awesome talk and also great demo. I did notice that in the video you showed, there are these artifacts that seem to become more intense as you move, and then they kind of resolve over time. Is that introduced by the SLM, or do you have insight on where those artifacts come from, and if that might induce a temporal limitation?
It's partially due to the SLM, so it's as you kind of tilt the angle, you might lose some light. Sorry. Another issue I'll note here is that when you change you put a displacement between these two elements, you shrink the aperture as well. So you end up blocking some light, which diminishes light intensity. We can predict that, and we can correct for that, in post, change the intensity in those regions if we wanted to. But it is, you lose some light in the system when you apply some focus.
I have a question. You show the Alvarez lens, Alvarez-Loman lens. You show only one cross section. I am assuming you have it's separable.
Yeah.
Are there any versions that are rotational symmetric?
That are rotationally
That
symmetric?
have some rotational symmetry.
So I'm showing this in 1D just for simplicity. Of course, these are 2D things, so you can do some fancy things. Typically, you move it in one direction, and that'll provide you some rotationally symmetric elements, but you can produce asymmetric elements as well, so you can make a cylindrical lens if you rotate it the other way. And that's actually pretty easy to do. You just have to put a different phase ramp on your SLM that's tilted in some other direction. So it's definitely possible. We didn't do too much with that yet, however.
Thank you. Okay, let's thank the speaker again. We are running on time. So our next speaker is Christopher Metzler. He's an assistant professor at the University of Maryland, and he has several awards, but I want to point to the fact that he has an NSF Career Award, an FOSR Young Investigator Award, and an ARO Early Career Award. So with that, I'd like to welcome Chris.
Well, thank you very much for the introduction. Obviously, I'm presenting here in lieu of Ashok Veeraraghavan. I'm going to talk about work that we've been doing together for the past 4.5 years or so now. And this was all done by our students, Brandon Fang, Haiyun Guo, Mingyang Ji, and Weiyun Zhang. Turbulence and scattering are arguably the two most important open problems in applied optics. Atmospheric turbulence is the central barrier to terrestrial astronomy. That's why we spent billions of dollars sending James Webb into space rather than building a much larger, cheaper telescope on the Earth. It's similarly a challenge for long-range surveillance applications, free space optical communications, and the related problem of scattering is a major issue in microscopy, where tissue limits how deep you can, scattering limits how deep you can see into tissue. It poses a problem for fiber-based multimodal endoscopy and non-invasive light-based pathology and screening. Fortunately, adaptive optics and wavefront shaping, in principle, solve these challenges. So recall that atmospheric turbulence is due to inhomogeneities in the atmosphere's index of refraction. That is, there's hot, low-density pockets of air where light passes relatively unimpeded, and cold, high-density packets of air where light slows down. And so as a wavefront passes through the atmosphere, it gets advanced and delayed, and you get a blurry and distorted image. The main idea behind adaptive optics is advance what was delayed, delay what was advanced to compensate for those aberrations, and go from something like you see on the left to something like what you see on the right, using deformable mirror arrays, SLMs, you name it. Similarly, a similar problem plays out in forward scattering. So there's a regime in scattering where you can essentially treat light passing through a scattering media as experiencing a phase delay. It's a very irregular, high-resolution phase delay, but that's sort of a useful simplification for the system. And just like with adaptive optics, if we put a spatial light modulator into the system, if we exactly delayed what was advanced and advanced what was delayed, we could recover a nice sharp image through the scene. And this is known as wavefront shaping, which is this adaptive optics with more degrees of freedom. So these systems, whether in the atmosphere or in tissue, work largely the same way, where you have a wavefront sensor, which actuates some adaptive optic system, deformable mirror array, or SLM, which after the wavefront's been measured, it's corrected, and you capture a nice sharp image. Unfortunately, almost all of these systems have an Achilles heel, and that is that by and large, they rely on guide stars. They rely on a few bright point sources in the scene that you can use to calibrate the system. And while this is okay in various astronomy applications where you can look up at the North Star or create something with a laser, this is a major issue in biomedical imaging applications where without really complicated tricks, it's hard to induce guide stars within tissue for calibration. Now, about 4 years ago, Ori Katz's group came up with what I consider sort of a breakthrough approach for imaging through scattering media, and that is image-guided wavefront shaping. And so the basic idea behind this approach is you have some object that you're imaging on the left, passing through some scattering media with a spatial light modulator that's conjugated onto the scattering media. So if you knew the phase error that was being induced by the scatter, you could put it on the SLM, conjugate it, and see through the tissue, see through the scattering media. Unfortunately, you don't know what that is beforehand. And so the approach they came up with is as follows. Let's put 100 random patterns on the SLM and capture 100 blurry and distorted images. And then for each of those images, compute some image quality metric. Is it sharp? Is it high contrast? Does it have high entropy? And then among those, take the best 5, create a new, 100 new patterns that are similar, put those on the SLM, take the best 5, capture 100 images, take the best 5 of those, so on and so forth. And gradually, sort of with this brute force genetic algorithm approach, optimize this metric to go from something like what you see on the top to like what the man you see on the bottom. And using this approach, they can correct severe optical aberrations, but it requires tens or hundreds of thousands of measurements. So it's pretty impractical. It's sort of incredible this works at all, but it's not useful for many applications. So the question we set out to solve is can we move keep this guide star free imaging through scattering capabilities while moving from 100,000 measurements down to just 100. And to do so, we combined we introduced neural wavefront shaping, which combines physical models with measurement modulation, statistical estimation theory, and neural signal representations to create a system which is passive, flexible, powerful, compact, and efficient, requiring only about 100 measurements compared to 100,000. And with that system, you can capture images of dynamic scenes that are optically corrected and look something like this. So the basics of the approach are as follows. You have the target that you're trying to image, the aberration, your lens, and without any correction, you get a blurry and distorted image. I'm going to describe this as if everything's static, but it very naturally extends to both dynamic objects and dynamic aberrations. Sort of optics 101, we treat that aberration just as a phase screen in front of the lens. And at this point, we essentially take the same optical system that Ori was using, where we have an additional SLM that we put a random pattern on that essentially applies an additional distortion to the imaging system. So now the measurements we capture have the form, each image is equal to the unknown object of interest, O0, convolved with some PSF, where this PSF depends on the unknown aberration phi, along with this known modulation gamma 1. And we capture another measurement of that form and another, so on and so forth. And you have a sequence of measurements where each of them depends on an unknown object, unknown aberration, but these known modulation patterns. And if you just assume that the noise is Gaussian, then we can just set up a maximum likelihood estimation problem. And if we could solve this, we could in principle reconstruct the object and reconstruct the aberration. Unfortunately, this is pretty challenging to solve in practice, particularly for high resolution objects and very strong aberrations. It's sort of a mix of both blind deconvolution. You need to separate the PSF from the object. You need the which is known. And then, once you have the PSF, you need to, solve a phase retrieval problem to reconstruct phi. And in fact, this is not a new formulation of imaging. This is known as phase diversity imaging. It's been around since the 90s to image through relatively mild optical aberrations. But you haven't been able to push the performance of the system until recently because it's so hard to solve that optimization problem. And fortunately, we have much better tools today to solve it, and in particular, we turn to neural signal representations. So, I'm assuming most of the audience is, at least vaguely familiar with neural signal representations. This is the same sort of idea that underlies neural radiance fields and other techniques. All it is that rather than representing an image as an array, we're going to represent it with a function, and we're going to use a neural network, a universal as a universal approximation function to represent that function. So, if you have, your neural network takes in pixel coordinates and spits out the intensity or the phase at that particular coordinate. So we parameterize our target and parameterize our aberration with these neural signal representations, and then take an analysis by synthesis approach where we simulate, if this estimate of the aberration was correct, if this estimate of the target was correct, what measurements would we produce, compare that with what we actually got, compute the likelihood, and then backpropagate and update your estimate of the aberration and update your estimate of the target. And with this technique, you can go from measurements that look like this and then reconstruct accurate estimates of the aberration and the target. And with the aberration and estimate in hand, you can perform adaptive optics, true adaptive optics, optical correction as well, by just putting the conjugate on your SLM. So we took this framework and applied it to a fairly simple experimental system where we have a resolution target imaged through various optical aberrations, in this case some glass covered in nail polish. And you get an uncorrected image that looks like this. But if you take this framework where you capture maybe 50, 100 measurements, you can reconstruct phase error estimates look like this, object estimates look like this, and take that estimate, optically correct to get something that's right at the diffraction limit of the system, and obviously much better than what you get without any correction. We applied this to various objects with various aberrations, onion skin, diffusers, nail polish. And it's really pushing the diffraction limit and almost pushing up against the diffraction limit in all these examples. But the real selling point of this approach, this neural signal representation approach, is that there's nothing necessarily unique about pixel coordinates. We can parameterize this with time as well. And so we can let our object estimate be a function of time, let our phase error estimate be a function of time, and reconstruct time-varying objects with time-varying aberrations. Here we have results with a varying aberration, static aberration with a varying object, and here we have dynamic object, dynamic aberration. So how much do these help? We could have taken that original formulation and tried to solve things in the first place. If you use a pixel-based representation, it just doesn't really work. You can get mediocre results with conventional subspace-based approaches, Zernike, for instance. But there's a huge advantage in using these representations due to their regularization, and they're really required to get any time-varying reconstructions. Sancho asked how many. So we went from 100,000 measurements to about 100 measurements in these examples. How many are really required? It ultimately depends on the strength of the aberration. So for pretty regular aberrations, we can get by with about 50 randomly chosen patterns. But for highly irregular patterns, we're requiring on the order of 500. And this shows up experimentally as well, where with 30 measurements, we're getting reconstructions like this, whereas for this stronger aberration, we might need 500. So we went from 100,000 aberrations down to 100. How do we get this down to 10? How do we get this down to something that almost interactive? So in what I described, the modulation patterns were chosen heuristically. We chose Zernike basis functions with randomized coefficients. You get modulation patterns that look like this. Can we do better if we design the modulation patterns? Can we choose the modulation patterns to get better corrections? And so here we turn to the end-to-end learning framework that Evan mentioned yesterday, where essentially we create a differentiable physical model of the optical system, pass training data through the system, model the reconstruction, and update the physical parameters. We applied this to the imaging through scattering problem. And what you quickly realize is that iterative algorithm, these neural signal representations we are using for the reconstruction, are too expensive for this approach to be super effective, and so we replaced it with a small proxy algorithm that allowed us to optimize these modulation patterns, choose the optimal patterns for imaging through the scattering, without having to rely on that big powerful algorithm for training. But then at inference time, we can take those learned patterns, apply them to an actual physical system, and then use whatever reconstruction algorithm we want, including the big iterative algorithms. And using these learned modulation patterns, we can go from something that looks like that to something that looks like that. And this applies to dynamic systems as well. So just the last minute or two of the talk, just want to briefly mention some sort of ongoing work on pushing this down all the way to two measurements. And this is based on an insight from Stanley Chan's group that asymmetric apertures enable fully computational wavefront sensing. So the basic idea is that, so let's ignore the scene for a second. Just imagine we're back to having a guide star, and we captured a PSF, and we want to estimate the phase error from the PSF. The insight that Stanley points out, based on some work that Jim Feenup and others did long before, was that there's sort of an inherent ambiguity associated with symmetric apertures. This phase error and this phase error produce exactly the same PSF, but if I correct with this PSF, this phase error instead of this phase error, I get completely different results. Whereas by contrast, if I have an asymmetric aperture, that conjugation and flip ambiguity disappears. This PSF and this phase error and this phase error produce completely different PSFs, and so you can reconstruct the PSF from the other. And so we combine this approach, this insight with a learning-based PSF estimation to perform guide star-free adaptive optics. So there's a trick in putting a high-resolution checkerboard pattern on an SLM, so you can use a phase-only SLM to perform amplitude modulation. But we can take these measurements, estimate the PSF with a trained neural network, estimate the phase error from the PSF with another trained neural network, put that conjugate pattern on the SLM, and then you can iterate this, sort of like a conventional adaptive optics system to remove residual aberrations as well. We applied this system to a range of different targets. So here you see, an uncorrected scene, and here's what the system looks like after correction. So in summary, the last 4 years, Ashok and I have been collaborating to really, push guide star-free adaptive optics systems. And in closing, want to thank all the students who made this work possible, as well as our sponsors. And shamelessly advertise, I have postdocs openings in my group if any of you are interested. Thank you.
We have time for questions.
Really nice, interesting talk. I have a few. I'll just do 2 questions, I guess. Is always physically separated from the aberrator. So I guess question number 1 is: would this work if your object was within, like, the cloud? Number 1, or within the aberrator? Like you're modeling your aberrating element as a 2D mask, I think, but in reality it's 3-dimensional, right?
Yeah. So
if your object was within that cloud, would there be any implications? And number 2 is, does this matter if you're limiting with coherent or incoherent light?
So all of these, I'll answer all of this is with incoherent light for right now. We're actively moving towards coherent light for sort of reflection mode imaging stuff, but don't have anything to show for that yet. And then revolumetric, I have some simulation results showing that you can compensate it. You can do sort of patch by patch reconstruction or some other stuff. But I think multi-conjugate is something we're interested in moving towards.
Any other questions?
Thanks. This is a super cool talk. So I have a question about you mentioned that you compared the neural parameterization of the phase mask against just pixel grid.
Yeah.
I'm curious if you tried, you mentioned that regularization was sort of what made the difference. I'm curious if you've tried just regularizing the pixel grid, like adding TV or stuff like that.
I think we tried TV, but I think the bigger selling point for us was that it captures temporal regularity. So yes, it improves the static one, but I don't know of a good way to parameterize like a video, if that makes sense.
Gotcha.
Other than INRs.
So time is an input to your...
Yeah, exactly.
Okay, cool. Thanks.
So I noticed that the models that you use to represent the effect of the aberrations seems to be a spatially invariant blur.
Yeah, so
right?
it's all isoplanatic right now.
So
yeah,
what
yeah.
kind of, could you sort of tell us what are the limits of that modeling approach? Like what kinds of scenes can you apply to?
Yeah,
if you shed some insight.
it's essentially the memory effect region. So however large the memory effect is, you can apply it there, and you can raster scan through potentially to image over a wider field of view. A lot of this, in theory, extends to multi-conjugate pretty trivially. In practice, it's super, super complicated. So I think that's something we're interested in, but yeah. And that would expand the field of view and deal with all those issues.
Okay, before we go, we have an announcement here, but let's thank the speakers, all the speakers again.
Okay, so as has been a tradition at ICCP, we typically take a group photo, and I think it would be a good time now, between now and the banquet, to just step outside to the courtyard and just do that right now. So we're all going to go to the area that's this grassy area that you can see from through the windows here. In the building. We're just going to go outside and do the photo over there. And also for the banquet, there should be printed QR codes with the Google Map directions to the banquet location. All right.